Metabolism is simply the process of managing energy in our cells. Metabolism is all of the chemical reactions that occur in the cell. Catabolism are the degradation reactions, the breaking large molecules into small ones. Anabolism are the synthesis reactions, building large molecules from small ones. So metabolism is simply the sum of our catabolism and our anabolism. Anabolism is simply making our needed molecules, making large molecules from smaller molecules. When we use amino acids to make proteins, that is anabolism. Catabolism is breaking down large molecules. Remember, energy is stored in chemical bonds. It takes energy to make the bonds in anabolism, and when we break the bonds, we're going to get that energy back out. Cellular respiration is the process of making ATP. We take the catabolism of our food fuels, primarily carbohydrates, but lipids and proteins can also be used, and when we break the bonds, we capture the energy and form ATP in our cells. Phosphorylation simply means adding a phosphate to a molecule. We phosphorylate ADP when we make ATP. We break off that phosphate from ATP, we get the energy to fuel a reaction, and we can stick that phosphate on another molecule. Phosphorylation is always enzyme-mediated. Oxidation-reduction reactions are paired reactions. Oxidation is when a molecule loses electrons. Now, since electrons are energy, when a molecule is oxidized, it loses energy. Reduction is the process of gaining electrons or energy. Because electrons are negative charges, it's not safe to let them travel alone, so we always make them travel with protons. Well, an electron and a proton is a hydrogen atom, so hydrogen atoms are transferred around as we're moving these electrons from molecule to molecule. Whenever we break bonds, we liberate energy, and we need to put that energy someplace. Whenever we break bonds or oxidize something, we liberate energy. We need to put that energy someplace pretty quick. So redox are simply the paired oxidation-reduction reactions that allow us to transfer energy from one molecule to another. You can remember oxidation reduction if you can remember Leo-Gur, lose electrons oxidation, gain electrons reduction, or oil rig. Oxidize is loss, reduction is gain. All redox reactions are catalyzed by enzymes. Dehydrogenases act as catalysts that move the hydrogens around. This means that we're moving those electrons, that's the energy we're moving around. Oxidases transfer oxygen. Coenzymes are special molecules that can temporarily hold hydrogen atoms. That is, they can hold those electrons temporarily. Most of these are obtained from vitamin D. FAD, or flavin adenine dinucleotide, and NAD, or nicotinamide adenine dinucleotide, are two very common ones we see in metabolism. These are their oxidized state. When they're reduced, they have picked up electrons, and we know that they've picked up electrons because these hydrogens have suddenly appeared. These hydrogens, remember, are carrying the electrons. ATP is synthesized in one of two ways, either substrate-level phosphorylation or oxidative phosphorylation. In substrate-level phosphorylation, the phosphate is on some substrate. We break the phosphate off the substrate and use the energy to transfer the phosphate to the ADP. This is the kind of energy that is used when we synthesize ATP in glycolysis and the Krebs cycle. The second way to form ATP is oxidative phosphorylation. This is a more complex process but yields a whole lot more ATP. The process is known as chemiosmosis. Electron energy is really kind of small, and chemiosmosis is a way to add up all of those little tiny electron bits of energy into enough energy to generate a lot of ATP. The electron energy is used to pump hydrogens across a membrane, and then we harvest the energy when they move back across the membrane. It's sort of like creating electricity with water and turbines. We dam up a river, and then we let the water flow and turn turbines. The turning turbines generate the electricity. We see oxidative phosphorylation in the electron transport system. So here you see substrate-level phosphorylation. We have an enzyme. All of these are enzyme mediated. Here's a substrate with a phosphate attached. This enzyme will break this bond and attach the phosphate to the ADP, and now we'll have ATP, that high energy bond in the ATP molecule, and the product here will no longer have phosphate on it. Oxidative phosphorylation, process of chemiosmosis, occurs because energy is released from the oxidation of food. Oxidation, remember, is breaking bonds and liberating electrons. That little bit of energy will pump hydrogen across the inner mitochondrial membrane. Remember, the mitochondria has two membranes, so we've got a space between the inner and the outer membrane, and we're going to pump a lot of hydrogens into that space. As a result, we're going to have a pretty steep hydrogen ion concentration gradient. Lots of hydrogens out here, almost none in here, and there are very few places where those hydrogens can come back across the membrane. Those places are these special enzymes called ATP synthases. When we get a buildup of hydrogen ions, we let them come through this port, and as they do, they generate the energy that will convert ADP and phosphate into ATP. The overall picture for carbohydrate metabolism is we take sugar, glucose, and some oxygen, and we end up making water, carbon dioxide, about 32 ATP, and some heat. There are three major processes in carbohydrate metabolism: glycolysis, the Krebs cycle, also known as the tricarboxylic acid cycle, TCA, or the citric acid cycle, and the electron transport system. All of this occurs in the cell. Glycolysis occurs in the cytoplasm. The citric acid cycle and the electron transport system occur in the mitochondria. Glycolysis and the citric acid cycle yield some ATP from substrate level phosphorylation. The electron transport system yields a whole lot of ATP from that oxidative phosphorylation. When glucose enters the cell, it enters by facilitated diffusion, and as soon as it's inside the cell, it's phosphorylated. It becomes glucose 6-phosphate. Most cells cannot let glucose 6-phosphate back out. They lack the enzymes to turn it back into just plain glucose, so glucose is trapped inside the cell. The other thing is because it's glucose 6-phosphate, it's not really glucose, so we manage to keep the intracellular glucose concentration low, and so glucose continues to enter the cell. Glycolysis literally means sugar splitting. We're going to take that glucose 6-phosphate molecule, which has 6 carbons, and we're going to split it in half. We're going to get 2 pyruvate molecules. Pyruvate contains 3 carbons. Now, because we break bonds, we're going to liberate some electrons. We have to put those electrons someplace, so we're going to reduce some NAD to NADH. Remember, we've got some electrons holding here that we can do something with later. We also managed to generate a couple of ATP out of substrate level phosphorylation. All of this occurs in the cytoplasm of the cell. While this is anaerobic, it doesn't use oxygen. Oxygen is present, we just don't need oxygen for this process to occur. Overall, we make 4 ATP, but there's startup energy. We have to invest 2 ATP in the process to get the ball rolling, so we say we only net 2 ATP. We're only 2 ATP ahead when we finish. Carbohydrates are the only molecule used this way. So here we see glycolysis, a 6-carbon glucose, ultimately broken down into 2 3-carbon pyruvic acids. Now there's a little step that has to go on, and as long as oxygen is present and we have plenty of NAD, we're going to go into the Krebs cycle. If oxygen isn't present, we can't generate enough NAD to run the Krebs cycle, and we'll end up making lactic acid. This is something that happens in your muscle cells. Now to enter the Krebs cycle, pyruvate has to go through a little intermediate step. The 2 pyruvate molecules will have a carbon each broken off of them. We'll get a 2-carbon acetyl-CoA molecule. Since we had 2 pyruvates, we'll get 2 acetyl-CoAs. Those carbons become carbon dioxide. Once again, we broke bonds, we liberated electrons, we're going to reduce some NAD. The acetyl-CoA goes into the Krebs cycle, and the Krebs cycle is nothing but a group of reactions that start with something, we rearrange carbons and break off some carbons, and we come back so that we can add acetyl-CoA again and start the process all over. It really is a cycle. Acetyl-CoA goes into the Krebs cycle. Since we have two acetyl-CoAs from each glucose molecule, what we get out of the Krebs cycle are 6 carbon dioxides. We do make 2 more ATP. We also break a lot of bonds and liberate a lot of electrons. We reduce some more NAD to NADH, and we reduce FAD to FADH2. This occurs in the mitochondrial matrix. This occurs on the inside of the mitochondria. The ATP made here is due to substrate level phosphorylation. We only get 2 ATP out of this, so far we have not made a whole lot of ATP from our glucose molecule. We need to harvest some more energy, and we have that energy stored in all of these NADH and FADH2 molecules that we have generated. Carbohydrates, lipids, and proteins can all enter the Krebs cycle at some point and can be used to generate ATP this way. Now here's the Krebs cycle. We take that pyruvic acid, break off a carbon and make carbon dioxide, and make this 2-carbon acetyl-CoA. This adds on to this 4-carbon oxaloacetic acid to make citric acid, and all we're doing is we just go around this circle, breaking off carbons, rearranging carbons, until we come back to this 4-carbon oxaloacetic acid, which can pick up 2 more acetyl-CoA, and we can go around again. So that's all this is. But notice that we break some bonds, so we generate some NADH, we generate a little ATP here, we generate some FADH2 and some more NADH here. So we store that electron energy in these electron carrier molecules, these coenzymes. The third step is the electron transport system. This occurs in the mitochondrial cristae. This is the space between the inner and outer membrane of the mitochondria. This is the only part of the reaction that absolutely must have oxygen. So this is an aerobic reaction. The reason we need oxygen is those hydrogen ions that we're generating have to have someplace ultimately to land. So oxygen is the final landing spot, and we'll make water. Once we make water, that's a very stable molecule. The ATP generated here will be generated by oxidative phosphorylation. We have all of those NADH and FADH2 molecules that we generated in glycolysis in the Krebs cycle. They're going to deliver their hydrogen atoms, taken from the glucose, to the cytochromes and flavins in the electron transport chain. Remember, those hydrogen atoms have the electrons for energy and the protons that will become the hydrogen ions we need later. As the electrons are passed from molecule to molecule in the electron transport chain, they lose a little bit of energy. Not very much, just a little bit. Just enough energy to pump hydrogen ions across that inner membrane into the space between the inner and outer membranes. We're going to build up a lot of hydrogen ions in the crista, in that space between the two membranes, and at some point in time we will let them flow back through the ATP synthase channel, and this will generate enough energy to produce ATP. Those hydrogen ions will combine with oxygen. We'll get H2O and we'll produce water. That will be one of our end products. This will yield approximately 28 ATP. So in the electron transport system, we have these various carrier molecules. You can see where NADH enters and where FADH2 enters. And as the hydrogen ions move from one molecule to another, at various places the electron is spit out. That little bit of energy is spit out. And that little bit of energy is just enough to pump one of those hydrogen ions across the membrane in the mitochondria. Once we get that buildup of hydrogen ions in the cristae, chemiosmosis will take over, and those hydrogen ions will flow through that ATP synthase channel, and this will generate enough energy that we can stick phosphates on ADP and make ATP. So the entire process, we have glycolysis occurring in the cytoplasm. We get 2 ATP out of this by substrate level phosphorylation, and we get 2 NADH that can go over here into the electron transport system. The pyruvic acid is converted to acetyl-CoA to enter the Krebs cycle, and the Krebs cycle occurs inside the mitochondria. We will generate 2 ATP from this, again by substrate level phosphorylation, but we will generate more NADH and FADH2. and all of these will go into the electron transport system. All of the NADH and FADH2 by the process of chemiosmosis will go through, and by oxidative phosphorylation will make about 28 ATP. So out of one glucose molecule, we can make about 32 ATP, with most of that being through the electron transport system. Cyanide is a metabolic poison, and the way cyanide works is it interrupts electron flow, and that causes metabolism to fail. As cells die, so do the organism. This is a very rapid-acting poison. Besides being broken down to make ATP, there are some other uses of glucose. Glycogenesis occurs when there is too much glucose and plenty of ATP, and we need to store that glucose. It's stored as glycogen, and this is stored in the liver. Glycogenolysis is breaking down that glycogen back to glucose so that we can get the glucose back and run it through carbohydrate metabolism to make ATP for energy. If there is an insufficient supply of carbohydrates in the diet, the body will turn to gluconeogenesis. Genesis, formation, neo-new, glucose. So this is forming new glucose. Here we're going to take proteins and fats, and we'll use them to make new glucose molecules. So gluconeogenesis is making glucose from non-carbohydrate sources. Glycogenesis and glycogenolysis balance each other to keep our blood glucose levels consistent. When we have plenty of glucose and plenty of ATP, we can convert that glucose to glycogen. If our blood glucose levels get low and we need ATP, we can break down the glycogen and turn it back into glucose that can go into the blood. You'll remember that lipids are highly concentrated energy storage molecules. In 1 gram of fat, there are 9 kilocalories of energy. In 1 gram of either carbohydrates or proteins, there are only 4 kilocalories of energy. Lipids are made of glycerol and 3 fatty acids. So when we break down lipids, we get glycerol and 3 fatty acids. Glycerol can become pyruvate and can enter that cycle for carbohydrates at the level of pyruvate. Fatty acids are long chains of carbon and hydrogen. We can break those carbons off 2 at a time. This is a process called beta oxidation, and turn them into acetyl-CoA, and that can enter the Krebs cycle. So when we have triglycerides, we can break them down into glycerol, get pyruvic acid, acetyl-CoA, go into the Krebs cycle, and the fatty acids can be broken down 2 carbons at a time. By that beta oxidation, we get acetyl-CoA, which goes into the citric acid cycle. Lipogenesis is the formation of triglycerides when we have to store that energy. When we have plenty of glucose and glycogen and ATP, glucose will be converted to fats. This is how we're going to store that energy. Lipolysis is breaking down that stored fat. This happens when glucose and ATP is low so that we can make more ATP. It turns out that fatty acids can directly be used by liver cells, cardiac muscle cells, and skeletal muscle cells to produce energy directly. We don't have to take it through the beta oxidation process. Metabolic acidosis is a condition that occurs when the body glucose levels are low and the body converts to gluconeogenesis, burning fats and proteins, and to lipolysis to obtain more glucose and more ATP. When those fatty acids are not completely oxidized, they build up as ketone bodies, and this makes the blood more acidic. Ketone bodies are acidic. People with ketoacidosis frequently have fruity breath. The acetone will vaporize, and that gives their breath a fruity odor. Metabolic acidosis based on glucose imbalance is common in older individuals, adults who are severely malnourished, people who go on those fad diets, and anorexics. The ketone bodies will be excreted in the urine and can be picked up on urinalysis. Acidosis means that the blood pH is low, and when blood pH gets low, this depresses the nervous system. Besides breaking fats down, we also have to use lipids to build things. We need lipids to build cell membranes, those phospholipids and the cholesterol. Lipids are not water soluble, so in order to transport them in the blood, we have to package them with proteins. These are called lipoproteins, and this is how we transport cholesterol and triglycerides in the blood. Cholesterol is synthesized in your liver. Most of the cholesterol in your blood is made in your liver. It does not come from your diet. Acetyl-CoA is what is required to form cholesterol, and cholesterol is essential for life. We need it to form bile salts, which helps us digest fats, and the steroid hormones, the sex hormones, testosterone, estrogen, and progesterone, and cortisol. And cortisol is vital for life. It is our stress hormone. Now here we see what happens. Here's our basic carbohydrate metabolism going on here. And you can see that acetyl-CoA can be used to make cholesterol for those steroid hormones and bile salts. If we build up a lot of acetyl-CoA because we have plenty of glucose, we'll drive the reaction this way. We can make fatty acids, we can make glycerol. Those will come together to make the triglycerides that are stored in the adipose tissue. On the other hand, if we need energy, we can go the other way. We can break the triglycerides down into glycerol and fatty acids by lipolysis. We can feed the glycerol in as pyruvic acid, and the fatty acids feed in as acetyl-CoA. And we can take those through the Krebs cycle and the electron transport chain to make ATP. If we are burning fats pretty exclusively, we're going to make those ketone bodies, and those ketone bodies are what lead to metabolic acidosis. Proteins are long chains of amino acids, and proteins deteriorate over time, so we're continually breaking proteins down and we continually need to replace them. Amino acids from deteriorated proteins are recycled. We can use those amino acids to make the new proteins or to make different compounds if we need to. If we take in more protein than we need, protein is not stored anywhere in the body. When our dietary protein is in excess, we have excessive amino acids. These are either oxidized for energy, they go into the Krebs cycle someplace, or they're converted to fat for storage. Proteins need to be replaced. We have to use those amino acids to replace degraded proteins. But if we're going to use them to make ATP, the first thing we have to do is deaminate them. The liver will remove that amine group, the part that contains the nitrogens, so that the rest of the molecule can be oxidized. The amines will be converted to ammonia and then excreted in the urine as urea. The remaining parts of the amino acid can enter the Krebs cycle at some point. Most amino acids are really used to synthesize new proteins. We don't really want to use them as a source of energy. We have to ingest 8 amino acids already formed. These are our essential amino acids. The others we can make if we have carbon, hydrogen, oxygen, and nitrogen, and enough energy to make them. We need proteins for enzymes, antibodies, and structures like hair and nails. The reason we don't want to break down proteins for energy is because it's not very efficient. It takes so much energy to convert the amino acid molecule to something that can go into the Krebs cycle that we frequently don't come out ahead on the ATP. So when we have too many proteins, we get the part of the molecule that's just the carbons, hydrogens, and oxygens, and we get the amine group. We have to deaminate that. So we make the urea, which comes out in the kidney, and the rest of the molecule is modified and can enter the citric acid cycle. So in our overview, here is how carbohydrates are used. This is straight through, and this is the way we want to make most of our energy, most of our ATP is using carbohydrates. We can detour excess carbohydrates to fats. We can break down fats to make ATP. We can also use some of the products here to make new amino acids, those we can make, but if we're breaking down proteins, there are certain amino acids that can fit in at various places here in the pyruvic acid, the acetyl-CoA, or even in the Krebs cycle. But it takes so much work to get them to this point that we usually don't make very much ATP from proteins.